When we are actually doing stoichiometry, we aren't just plugging numbers into a calculator to get an answer. We are doing an experiment in a lab to determine the amount, whether that be reactant, product, or unknown. The general term for that would be a quantitative analysis. Very often, we will do a reaction, maybe form a precipitate, and mass that precipitate. A general term for that sort of process where we are analyzing a sample by mass is gravimetric analysis, a measurement using gravity. Titration is a common process used with aqueous solutions to analyze the concentration or other composition of an unknown. It involves careful measurement of solutions and a very precise reaction to find out our unknown amounts. In a titration, we use a piece of glassware called a burette, represented here, here, and here, that precisely delivers volume of solutions. There is a stopcock or a valve down here at the bottom that we can open a little at a time to allow single drops, much less than a milliliter of a solution, to fall in. And by carefully measuring that and comparing it to predicted changes, we can figure out something about the unknown. The solution in the burette is called the titrant, and it's added to the analyte. That's usually our unknown, and our unknown is either measured by mass or carefully measured by a volumetric pipette until we know exactly how much is there. So we may have measured out an unknown. Sometimes we use an indicator or other tools to let us know when our reaction is complete, and then we add our analyte. What you see here is a solution that changes colors when we get to a certain amount. We have an initial reading, we add liquid, then we have a final reading. The right amount of titrant being added is called the equivalence point. That's when we've added the correct stoichiometric amount of that chemical to react with the amount of analyte that we have. The visual evidence we might be looking for would be a color change of a solution, maybe caused by the reaction itself, a color change of an acid-base indicator that tells us about a pH change, a pH change that we observe using a pH probe or meter, the formation of a precipitate, maybe bubbling, or even change in electrical conductivity. There's lots of different signs that we can use, but they're all based upon knowing that we have added a certain amount of one chemical to the other. The visual evidence that we are done is called the endpoint. When we get to that endpoint, maybe the change in color, we measure. So the equivalence point is where, according to our mole ratio, we have equivalent amounts of each thing so that they fully react. The endpoint is our visual evidence that something has changed. And obviously, we need those 2 points to match up. Sometimes that's a challenge with certain reactions, so we're very careful in choosing our method. We will talk about acid-base indicators and pH changes in much more detail in the acid-base equilibrium unit, and we can even talk about changes of color in the redox unit. But when we're doing these calculations, what we think about is we would know the molarity of our titrant, because that would be our known solution. We would also be able to carefully measure the volume of that. By knowing the molarity and volume, we determine the moles of what we added. From our balanced chemical equation, we use our mole ratio. So based upon the moles of chemical we added, we can figure out how many moles of chemical were down here. Then we connect that to either our desired value, maybe a molarity, thinking about volume and moles, maybe a mass or a molar mass. By carefully measuring this at the beginning in terms of volume or in terms of mass, we can make the connection and the calculation that we need to. Really, titrations are just the application of a specific type of stoichiometry. There's almost always going to be 1 to 2 uses of the molarity. We need to remember to look at our mole ratio, and then we need to be very careful about what our final mole value is. The last thing to recognize here is that the molarity and volumes are very precisely measured. On our burette, we are measuring volumes. And when we have a burette that looks like this with a 28 mark and a 29 mark with 10 in between, that means not only can we measure to the nearest milliliter, we would estimate between the smallest markings. So this wouldn't be 28 or 29. It wouldn't be 28.56778. It would be between 28.8 and 28.9, something like 28.85 or 28.86. The way that a burette is numbered is very unique. You have to look at the fact that it's numbered from a 0 mark down to a 25 or 50. You do not need to do any further calculation when thinking about the volume or recording the measurements. You record exactly as presented, and then by doing a subtraction, you figure out how much is added.